Imagine a world where ancient humans and Neanderthals roamed side by side, their lives intertwining in ways we're only beginning to understand. Now picture a group of people in northern Spain and southern France, the Basques, whose blood and language set them apart from everyone else. Could there be a hidden link between the Neanderthals' unusual blood traits and the Basques' high rate of Rh-negative blood? It's a question that pulls us into a mystery spanning tens of thousands of years, blending genetics, history, and a dash of speculation. Let's dive into the details of Neanderthal Rh plus incomplete blood, the Basque Rh-negative puzzle, and whether interbreeding might have sparked a genetic twist that echoes through time. What's so special about Rh-negative blood? First, let's get the basics down. Your blood type isn't just about A, B, or O, it's also about the Rh factor, a protein on red blood cells. If you have it, you're Rh positive. If you don't, you're Rh negative. About 85% of people worldwide are Rh positive, but in parts of Europe, especially among the Basques, Rh negative is far more common. Up to 35% of them lack this protein. That's a huge jump from the global average of 15% or the near-zero rates in Asia and Africa. Why are the Basques so different? Scientists think Rh-negative comes from a mutation, a deletion in the Rhd gene that stops the protein from forming. But when and where did this mutation start, and could Neanderthals have anything to do with it? Neanderthals, our extinct cousins who lived across Europe and Asia until about 40,000 years ago, had their own twist on blood types. Thanks to ancient DNA pulled from bones in Spain and Russia, we've learned they weren't all plain Rh positive like most modern humans. Some had what scientists call Rh plus incomplete. So what does that mean? Unlike the standard Rh positive, where the Rhd gene pumps out a full-strength D antigen, that protein, this incomplete version makes a weaker or slightly different one. It's still there, so they're technically Rh positive, but it's not the same old, same old. Picture it like this. If regular Rh positive is a bright red light, Rh plus incomplete is more like a dim orange glow, still on, but funky. Researchers found this in Neanderthal DNA through specific changes in the Rhd gene, which tweak how the protein works. One Neanderthal from Spain's El Sidron cave had this oddball trait, and it's super rare in humans today, showing up only in 2 people, one Aboriginal Australian and one Papuan. That rarity hints it might have slipped into modern humans from Neanderthals, but could it have messed with human blood in a bigger way, like flipping it to Rh negative? We know Neanderthals and early modern humans hooked up and DNA swapped hands. Today, Europeans carry up to 2% Neanderthal genes, think skin color tweaks or immune system boosts. The Basques, tucked in their mountain corner, have a bit more, around 2.5 to 3%, suggesting they kept more of that ancient mix. Could this interbreeding have stirred the pot for something as specific as Rh-negative blood? One clue comes from a guy named Oase1, a 40,000-year-old human found in Romania. His DNA shows he had a big chunk of Neanderthal genes, up to 9%, way more than modern folks. That means his ancestors got cozy with Neanderthals just a few generations back, maybe 50,000 years ago. Oase1 lived in Eastern Europe, not Spain, but it proves interbreeding left fresh marks in human genomes right where Neanderthals roamed. If it happened there, why not in Spain, where Neanderthals might have hung on longer? Did Neanderthals stick around in Spain? Here's where it gets juicy. Most experts say Neanderthals vanished by 40,000 years ago, but some bones from southern Spain, like at Gibraltar's Gorham's Cave, hint they survived until 28,000 or even 25,000 years ago. That's late, way past the usual cutoff. If true, it puts Neanderthals in Spain overlapping with modern humans longer than we thought, especially in a cozy refugium where the Ice Age wasn't as brutal. The Basque region isn't far from these spots. Could late meetups have left a genetic fingerprint like Rh-negative? How does Rh-negative happen? Rh-negative comes from a broken Rhd gene. Specifically, a chunk of it gets deleted, so no D antigen shows up. You need 2 copies of this busted gene, one from each parent, to be Rh-negative. Scientists guess this mutation popped up over 100,000 years ago in Africa, since it's there in tiny amounts, 1 to 3%. But it really took off in Europe, hitting high notes in places like Spain's Basque country. Some estimates peg its rise to 30,000 to 35,000 years ago, based on genetic drift or maybe a survival perk we haven't nailed down. But what if it's older, like 50,000 years ago, and tied to Neanderthal mixing? Think about blue eyes. We used to assume they were recent, maybe 10,000 years old, but ancient DNA from a 19,000-year-old Italian showed they go back further, possibly 20,000 or even 50,000 years. If blue eyes are more ancient than once believed, could Rh negative be too? Maybe it simmered in humans earlier, waiting for the right moment or the right interbreeding to spread. Could Rh incomplete spark Rh negative? Now let's wrestle with the big question. Could Neanderthal Rh plus incomplete have messed with human genes to create Rh negative? Imagine a Neanderthal with rhesus incomplete hooking up with a human carrying the standard Rh positive. Their kid might get a mix that's still Rh positive, just with a quirky antigen. But what if something weirder happened? Could that mix-up somehow break the Rhd gene entirely, flipping it to Rh negative? Rh negative isn't just a tweak, it's a deletion, a clean cut in the DNA. The Neanderthal rhesus incomplete still makes an antigen even if it's weak. Mixing it with human Rh positive doesn't naturally erase the gene. It might make a funky hybrid, but not a total knockout. For Rh negative to pop out, you'd need both parents passing on the gene, and Neanderthals don't show that deletion in their DNA, at least not in the few we've checked. But what if we're missing something? Only a handful of Neanderthal genomes are sequenced. If some in Spain carried a hidden Rh negative allele, or if their rhesus incomplete messed with human genes in ways we don't get yet, could interbreeding have sparked a mutation? OACE1's heavy Neanderthal load shows how fast their DNA could flood into humans. Maybe in Spain, 50,000 or 25,000 years ago, a similar flood flipped a switch. The Basques make this idea tempting. Their Rh negative rate is sky high, and they've got that extra Neanderthal DNA kick. Plus, they're near where Neanderthals might have lingered. Picture this. Modern humans in Spain 40,000 years back meet Neanderthals. They interbreed, and over generations, something shifts. Maybe the mutation doesn't directly cause Rh negative, but the genetic chaos of mixing triggers a mutation, a deletion, that sticks around. By 25,000 years ago, as Neanderthals fade, these humans, proto-Basques, are left with a rare trait that grows in their isolated mountain home. Why isolation? The Pyrenees kept the Basques apart from later waves, like Indo-European farmers who swept Europe 5,000 years ago. That separation preserved their weird blood and their language, Euskara, which doesn't match anything else in Europe. Could Neanderthal contact have set the stage for both? What's the genome telling us? Let's zoom into the DNA. OACE1's case shows Neanderthal genes pouring into humans fast and thick, 9% in one guy, just 40,000 years ago. If that happened in Spain too, it's not crazy to think it shook up the Rhd gene. Modern Basques have Neanderthal snippets scattered across their genome, and while we haven't found the D allele in Neanderthals, we've only peeked at a few. The L Sidron Neanderthals' partial D proves their blood was diverse. What if others had something closer to Rh negative? Compare that to Africa, where Rh negative is rare. If it started there 100,000 years ago, why didn't it spread? Maybe it needed a boost, like Neanderthal interbreeding in Europe, to take off. The timing fits. 50,000 years ago, humans hit Europe, met Neanderthals, and genes swapped. By 30,000 years ago, Rh negative could have been brewing, hitting high gear in places like Spain. Here's another angle. Immunity. Rh negative moms carrying Rh positive babies can have trouble. Her body might attack the fetus's blood, causing hemolytic disease. If Neanderthals were mostly Rh positive, and some human mums were Rh negative, hybrid pregnancies could have been rocky. Did that pressure nudge Rh negative to rise as a way to avoid mismatches? It's a stretch, but maybe in small groups it flipped the odds. Neanderthals might have added a wrinkle. Their weak antigen could have confused human immune systems, sparking odd reactions. Could that chaos have broken the gene in some offspring, leading to Rh negative? It's not proven, but it's a wild card worth pondering. But then there's the Basque hook, their blood, their language, their Neanderthal ancestry. It's a perfect storm of clues. Euskara, the Basque language, is a linguistic enigma, a stubborn survivor in a sea of Indo-European tongues like Spanish and French. Spoken today by about 700,000 people in northern Spain and southwestern France, it's a language isolate, meaning it has no known relatives. Its oddball grammar, unique words, and ancient vibe have long puzzled scholars. But what if Euskara isn't just a human relic? What if it carries echoes of Neanderthals, our extinct cousins who roamed Europe until at least 40,000 years ago, and maybe later in Spain? Let's explore Euskara's quirks and speculate whether it could be a Neanderthal remnant. What makes Euskara stand out? Euskara doesn't play by Europe's usual rules. Most languages around it, Spanish, French, English, trace back to Indo-European roots, brought by farmers 5,000 to 7,000 years ago. Euskara doesn't fit that mold. It's agglutinative, stringing suffixes onto words like beads on a thread. Etixi, house, becomes etixen, in the house. Its grammar is ergative, flipping how subjects and objects work compared to I run or I hit the ball. Words like geison, man, or water, don't match Latin, Celtic, or Germanic cousins. It feels old, really old, like it's been hiding in the Pyrenees forever. Could that forever stretch back to Neanderthals? Neanderthals weren't mutes. They had a version of the FOXP2 gene linked to speech in humans, and their hyoid bone, a throat bone for talking, looks like ours. Evidence from tools, burials, and art suggests they communicated maybe with sounds, gestures, or even basic words. But complex language like ours, with syntax and storytelling? That's trickier to prove. Most experts say their speech, if it existed, was simpler, grunts and short phrases, not poetry. So, could Euskara, with its intricate grammar, come from them? Not directly. But what if it's a language shaped by Neanderthal contact? Indeed, Neanderthals might have survived until 25,000 years ago. The Basques, Euskara's speakers, carry 2.5 to 3% Neanderthal DNA, slightly more than most Europeans. What if, during those late meetups, humans picked up bits of Neanderthal communication? Imagine early humans in Spain, trading words or sounds with Neanderthals over a campfire. Euskara might have borrowed quirks, like its choppy rhythm or odd structure, from Neanderthal grunts evolving into something unique over millennia. Spain's a hotspot for this idea. Neanderthal sites like El Cedron near Basque turf show they hung around. The Pyrenees later shielded Basques from Indo-European waves, preserving their language and genes, including high Rh-negative blood, another Neanderthal-tinged mystery. If Neanderthals had a language, Euskara could be its fossil, polished by humans, but rooted in that ancient mix. Its isolation kept it from blending away like a time capsule. Euskara as a Neanderthal remnant is a long shot. Neanderthal speech likely wasn't complex enough to birth it outright. But as a hybrid, tweaked by contact? Maybe. It's a tantalizing what if, a language born in human throats but flavored by Neanderthal echoes, surviving where others faded. Could those ancient grunts still hum in Basque hills? Maybe Rh-negative didn't start with Neanderthals, but got a push from them, like a seed sprouting in the right soil. So could Neanderthal Rh plus incomplete tie to Basque Rh-negative blood? It's not a straight shot, but if Neanderthals hung on in Spain until 25,000 years ago, interbreeding with humans 50,000 or 30,000 years back could have stirred the genetic pot. OACE1 proves Neanderthal DNA hit humans hard and fast. Why not in Spain, boosting a mutation like D? Maybe it's not the origin but the amplifier, turning a rare African quirk into a Basque hallmark. What do you think? Could a Neanderthal kiss have left us with Rh-negative blood, or is it just a coincidence of timing and place? The Rh null blood type, also known as golden blood, is one of the rarest blood types in the world, and it carries a unique mystique within the field of science. This exceptionally rare blood type lacks all Rh antigens. Fewer than 50 known people worldwide are thought to possess this blood type, and the study of this blood type reveals fascinating insights into human genetics and population isolation. The first case of golden blood was discovered in an indigenous Australian woman. Since then, dozens of cases have been discovered, but scientists estimate that it affects approximately 1 in every 6 million people worldwide. Nobody knows for certain, but there have only been 43 confirmed cases. Blood type is inherited, and Rh-null blood is no exception. It is caused by a mutation in the RHCE gene inherited from both parents, a rare phenomenon known as autosomal recessive inheritance. This occurs when a person inherits 2 copies of the mutated gene, one from each parent. The indigenous populations of Australia and Papua New Guinea have been relatively isolated for tens of thousands of years. This isolation has allowed rare genetic traits, including the Rh-null blood type, to persist in higher proportions than in more genetically diverse populations. Indeed, Rh-null blood has been disproportionately observed in individuals from indigenous communities in Australia and Papua New Guinea. This is likely due to genetic isolation and the unique genetic makeup of these populations. As a matter of fact, the genetic makeup of isolated communities often reflects that of their early ancestors. If the small founding populations carried the mutations associated with Rh-null blood, it could have become more common over generations. Smaller gene pools in isolated communities make it more likely for recessive traits like Rh-null to appear in individuals when 2 carriers of the gene have offspring. The rhesus blood system, named after the rhesus monkey, is one of the most complex blood group systems, with about 61 different antigens on the surface of red blood cells. Most people have some combination of these antigens, classified as Rh positive or Rh negative. However, Rh-null individuals lack all Rh antigens, a complete absence that sets them apart from any Rh positive or Rh negative blood type. The Rh-null mutation is believed to have arisen independently in different parts of the world, but remains incredibly rare due to the combination of genetic factors needed to produce it. Rh negative and Rh positive blood types are determined by the presence or absence of a protein known as the rhesus factor on the surface of red blood cells. Roughly 85% of the world's population carries this protein, making them Rh positive, while the remaining 15%, primarily found in Europe and parts of North Africa, are Rh negative. The origins of the Rh negative mutation remain a mystery. Scientists estimate it appeared thousands of years ago, although exactly when and where is still up for debate. Genetic studies show that Aboriginal Australians have one of the oldest lineages outside of Africa, with evidence suggesting they reached Australia between 50,000 and 65,000 years ago. For Australian Aboriginals, their unique genetic makeup reflects a lineage that remained largely unchanged until relatively recent contact with other populations. This isolation contributes to their distinct set of genetic traits, including a high frequency of Rh-positive blood and the rare Rh-null variant. In a world where every blood type tells a story, golden blood remains an enigma, illuminating the connections between populations and the mysteries of human genetics. Through the study of rare traits like Rh-null blood, we gain a deeper understanding of the intricate puzzle of human diversity, where each piece plays a vital role in the survival and adaptability of our species. However, unlike some other populations around the world, Australian Aboriginals almost exclusively possess the Rh-positive blood factor, making them a unique case in the study of human genetics. What does this tell us about their ancient migration patterns and how isolated their populations have remained over tens of thousands of years? The blood type distribution among Australian Aboriginals is one of the unique genetic traits that highlight their isolation and ancient ancestry. Unlike some other populations, Australian Aboriginals predominantly have Rh-positive blood types, with Rh-negative types being extremely rare. This pattern differs from certain European populations, where the Rh-negative trait is more common and has been linked, speculatively, to Neanderthal ancestry. Nonetheless, Australian Aboriginals' predominantly Rh-positive blood suggests limited influence from Neanderthal genes, specifically tied to Rh-negative traits. One of the most remarkable aspects of Aboriginal genetic history is its continuity. Aboriginal Australians are believed to have settled on the continent between 50,000 and 65,000 years ago, making them some of the earliest known migrants out of Africa. This isolation is likely the reason Aboriginal Australians have such a distinct genetic profile, with relatively little mixing with other populations until recent centuries. The absence of Rh-negative blood among Aboriginal Australians aligns with the theory that this genetic trait arose later, likely after their ancestors had already migrated to the Australian continent. The Rh-negative mutation appears to have emerged primarily in European populations, which would explain its near absence among Aboriginal Australians, as well as other indigenous groups with long-standing isolation, including Native Americans. The story of Rh-negative blood in global populations provides clues about ancient human migration and separation. Australian Aboriginals are unique in that their ancestors likely branched off from other early human populations but did not come into contact with European populations where the Rh-negative mutation became prevalent. This genetic isolation supports the hypothesis that Australian Aboriginals represent a direct lineage from some of the earliest modern humans. As these groups travelled across Asia and eventually reached Australia, their paths diverged from other populations who moved toward Europe, where the Rh-negative blood trait became more common. The extremely low incidence of Rh-negative blood in Australia reflects the absence of any significant gene flow from populations in Europe, Central Asia, or other regions where Rh-negative genes are more prevalent. While the isolation of Aboriginal Australians explains the absence of Rh-negative blood, some alternative theories propose a more complex prehistoric migration story. These theories, such as the lost migration hypothesis, suggest that small populations might have crossed parts of the Indian Ocean or followed coastal routes from Africa or South Asia. This hypothesis has led some to wonder if Rh-negative blood would have appeared in the gene pool had any of these migrations been more extensive, or if the mutation itself is simply a more recent development in human evolution. Whatever the case, the almost exclusive presence of Rh-positive blood among Australian Aboriginals raises intriguing questions about early human evolution and migration. Why did the Rh-negative mutation emerge primarily in Europe and not in other parts of the world? Is the lack of Rh-negative blood among Aboriginal Australians simply a matter of timing and isolation, or could there be unknown factors that influenced the trait's distribution across ancient populations? Australian Aboriginals' unique genetic makeup is a living testament to an ancient human journey that began tens of thousands of years ago. The absence of Rh-negative blood is more than a genetic trait. It's a clue, helping us piece together a picture of prehistoric migrations, isolation, and survival. In a world where most populations exhibit a mixture of genetic influences, the near-complete absence of Rh-negative blood among Aboriginal Australians is a reminder of the profound impact of isolation on the human genome and of the resilience and continuity of one of the world's oldest civilizations. Australian Aboriginals likely acquired different genetic adaptations during their ancient migrations, which shaped their unique blood type distribution. They may have encountered Denisovans, another group of archaic humans, rather than Neanderthals directly, given the geographic locations where these populations lived. Genetic studies show that Aboriginal Australians and Papuans carry more Denisovan DNA than Neanderthal DNA, which could explain the rarity of the Rh-negative trait in Aboriginal populations. While Australian Aboriginals have some Neanderthal ancestry, they also carry a notable amount of Denisovan DNA. Denisovans were another group of archaic humans whose remains have been found in Siberia and Southeast Asia. Genetic analysis reveals that Denisovans contributed genes to Melanesian, Aboriginal Australian, and some Southeast Asian populations, providing adaptations that may have helped them survive in various environments. In fact, one study suggests this interbreeding occurred as recently as 15,000 years ago in Papua. Denisovan genes in Aboriginal Australians may have influenced traits like immune response, height, and potentially some skeletal features. For example, it's plausible that they may have influenced Australian Aboriginals in ways suited to the Australian climate. While the Denisovan contribution to blood types is not fully understood, these ancient genes add a layer of complexity to the genetic tapestry of Australian Aboriginals, further distinguishing them from other populations. The connection between Australian Aboriginals and Neanderthals and Denisovans tells a story of resilience and adaptation. Their predominantly Rh-positive blood type, unique genetics illustrate how Aboriginal Australians developed independently after their ancestors arrived on the Australian continent tens of thousands of years ago. Though their Neanderthal DNA contribution is relatively low compared to European populations, their Denisovan ancestry reveals a rich legacy of ancient genetic adaptations that shaped their physiology and biology. The legacy of Neanderthals and Denisovans in Australian Aboriginals is a testament to the vast and complex history of human evolution. While Australian Aboriginals took a path that led them to thrive in the challenging landscapes of Australia, their ancient genetic ties offer us a glimpse into the interwoven stories of humanity's journey out of Africa and across the continents, where traces of archaic humans endure in the diverse characteristics of modern people. A final note, commenting that Aboriginal Australians don't have any more claim to be native to the Australia fails to consider the unique relationship Indigenous people have with their lands based on thousands of years of uninterrupted presence, stewardship, and cultural connection. Thus, arguing that Aboriginal Australians aren't native because their ancestors migrated there is an illogical argument. Aboriginal Australians have profound, continuous, and culturally specific connections to their lands that go far beyond the simplistic idea of first arrival. In the end, the story of NULL blood may be just one thread in the larger tapestry of human history, but it's one that connects us to a distant, mysterious past, one that continues to fascinate and inspire researchers and storytellers alike. Now we leave you to ponder the mysteries of our shared human history. Thanks for watching, sharing and subscribing, and take care.